Hello, everyone. Thank you so much for coming to this month's PK University, where we will be hearing about the 2025 NPKUA Research Grant projects. These were the projects that were awarded this year through our annual grant program. So you'll get to watch quick videos about the summaries of each project. And then we also have all the grant awardees here live to answer any questions as they come up. So we're very thankful for them taking the time today to do this and very thankful for their work for NPKUA. And just to give a little background on our grant program, as I mentioned, this is an annual grant program we've had since 2010. And since then, we have funded over 85 projects for nearly $6.5 million toward PKU research. So this is a program we're very proud of, and we're really grateful for the researchers today to be able to share. So with that, we will go ahead and go into our first one. So the first grant project we'll be hearing from is from Dr. Carrie Harding.
I'm Dr. Carrie Harding, and it's my pleasure to present to you on our research project that's funded by the National PKU Alliance. It's entitled Novel Approaches to Achieving Permanent Therapeutic Gene Insertion in PAH-Deficient Mice. In our lab, our goals are to contribute to the overall understanding of PKU, to develop new genetic therapies for PKU, and to try to better the lives of individuals who are living with PKU. We have a very, very small team of researchers involved in this effort here at Oregon Health and Sciences University. As you all know, the problem in PKU is that both phenylalanine hydroxylase, or PAH genes, in an individual with PKU carry variations that lead to the production of an abnormal messenger RNA. And this leads either to dysfunctional PAH protein or the complete lack of PAH protein, and therefore the inability to convert phenylalanine to tyrosine. And for many years, we've worked to develop gene therapy methods that will correct this problem in liver. In recent years, we've focused upon using gene editing techniques to directly alter DNA in liver of PKU mice to permanently restore PAH enzyme production and therefore lower their blood Phe levels. We started out working simply to correct the gene variant that causes PAH deficiency in one of our PKU mouse models. The outcome of this effort was similar to a technique called base editing that you might have heard about. Earlier this year, base editing in liver was used to partially treat a baby boy with a different rare metabolic disease that also involves protein metabolism but is in the urea cycle. The problem is that we now know of more than 1,600 different specific variants or specific changes in the phenylalanine hydroxylase gene that are known to cause PKU worldwide. And since individuals with PAH deficiency each have two variants, there are actually over 3,600 different combinations. Some variants, like the variant that changes amino acid 408 from arginine to tryptophan, are somewhat more common than other variants and may be particularly common in certain specific geographic regions. But no single variant causes the majority of cases of PKU. And there are gene editing techniques available that can correct individual gene variants, but we'd have to essentially design 1,600 different drugs to treat the entire world of PKU. So this is a daunting problem. For several years now, we've been exploring methods that will treat anyone, regardless of what PAH variant they've inherited. Principally, the gene editing methods we're developing aim to insert a new copy of the entire PAH gene at some safe place in the liver DNA and start making PAH enzyme. We've had some success using an enzyme system called CRISPR-Cas9. Cas9 is an enzyme that's native to many bacteria. For bacteria, it's part of their defense mechanism against foreign invaders, namely viruses that like to infect bacteria. And Cas9's function is to locate and destroy foreign DNA. It's very discriminatory. It seeks out very specific DNA sequences and cuts the DNA. The beautiful thing about Cas9 is that it can also be programmed to precisely locate and cut specific sites in mammalian DNA as well. We use viral gene therapy, specifically adeno-associated virus or AAV, to deliver the necessary reagents to produce Cas9 in liver. We've engineered the system to cut in the mouse PAH gene and to cut only there. Now, Cas9 will frequently cut both chromosomes, but I'm just showing it cutting one. Now, Cas9 just makes a cut and leaves a hole. And somehow a lot of people, even scientists, have misunderstood that CRISPR-Cas9 editing can cure disease. But that mostly isn't true for recessively inherited disorders. It just makes a cut in the DNA. It doesn't fix anything. The liver cell does not like having a cut in the DNA, so it will immediately try to fix the cut using one of a few different repair methods. It can just put the two ends of the DNA back together again, but that's not useful to us. We introduce a second AAV vector that carries a healthy PAH gene. And what we hope for is that the liver cell will incorporate this new DNA fragment, what we call a repair template, into the cut. And if this is done correctly, the liver DNA now carries a copy of the healthy PAH gene and will start producing PAH enzyme. We've had some success with this approach, but this only works if we treat the mice when they're infants. The mouse on the right here was treated with the appropriate AAV vectors when it was a pup. And now as an adult, its blood Phe, although still elevated, has come down to about 400 micromolar in comparison to a mouse in the middle, which was not treated. High blood Phe partially blocks production of melanin, so that changes the coat color of a genetically black mouse. And we can see that the treatment has worked pretty well since the animal's coat color is now black instead of tan. However, not all of the mice that are treated are corrected this well, and we just simply want to do better. For our next phase of research, we've been working to adapt a new technology called PASTE, which stands for Programmable Addition via Site-Specific Targeting Elements. And this method was first published in 2023 by a group at the Massachusetts Institute of Technology. I will spare you the details, but PASTE is a complicated version of CRISPR-Cas technology. It still uses the Cas9 enzyme, which is shown here as part of this enzyme complex on the bottom. And Cas9 is still used to locate and cut the liver DNA at a specific site, but the system includes two other enzymes, which are at the top here. And these two other enzymes literally paste in a new gene at the place that Cas9 makes a cut. We propose that this system will be more successful than our previous method in achieving PAH gene insertions in a greater number of liver cells. And we've planned a series of experiments to test this. Also, we think this technique will work in both adult and juvenile mice. We propose that successful gene therapy for PKU will permanently reduce blood Phe levels, reduce or eliminate the need for the low Phe diet, and ultimately will lead to healthier lives overall. We profusely thank the National PKU Alliance for their continued support of our efforts. All of our gene editing work has been funded by philanthropy and would not have been possible without financial support from NPKUA. And I thank you very much for your attention today to our presentation.
Okay, great. Thank you, Dr. Harding, for that video about your grant project. Now we have time for questions about his grant. And Dr. Harding, unfortunately, was unable to join us today, but Dr. Laura Young from his lab is here to answer questions in his place. So if you have any questions, please put them below in the Q&A box. Okay, it looks like we have one. So what about direct AAV delivery of the PAH gene?
Is we're trying to integrate it into the genome so that it has a withstanding therapeutic effect. Because if you just add the transgene copy into the liver cells, the liver likes to turn over, it likes to replicate, and eventually that will get lost. And so it wouldn't have a lasting therapeutic effect. So that's why we have to have the addition of the Cas9 gene editor.
Great. Well, thank you. Well, just to make sure we stay on time, we're going to move to the next one, but if we have time at the end, we can go back and answer some of the other questions. So thank you. So next is Dr. Charlotte Lubit. So I will play her video.
Good afternoon. My name is Charlotte Lubbout. I'm a pediatrician metabolic diseases in the Netherlands. And during this presentation, I would like to tell you about our AI for PKU study. But first, I would like to thank the National PKU Alliance for funding our research and making our AI for PKU study possible. Many thanks. So let's first meet our study team. Our study team is located in the Netherlands, a country in Europe. We have two study centers located in the south, Nijmegen, and in the north, Groningen. We have a multidisciplinary study team with metabolic dietitians, metabolic pediatrician, a clinical chemist, metabolic diseases who is specialized in artificial intelligence, a nurse specialist, and a medical student who became enthusiastic about doing research on PKU after her first year's lecture on PKU. So together with our experience in treating PKU, we will start this study. The background of our study is that in infants with PKU, phenylalanine should be in range soon. And often the baby is diagnosed by newborn screening, and then phenylalanine can be already very high. So then the PKU diet is started to get phenylalanine down. And this diet consists of phenylalanine-free formula, often combined with a little bit of normal milk, that can be mother's milk or normal formula. And it can take some time to have the diet that gives a good phenylalanine value in range, and often many adjustments need to be done. The question for our study is, can artificial intelligence help us with getting the right diet in babies with PKU? And for this, it is important to realize what AI, artificial intelligence, is. Nowadays, it's everywhere. And you can see AI like a smart computer program that can learn based on a lot of information, a lot of data, and make decisions just like people do. It looks at information, finds patterns, and then uses that to help us. For instance, what television program we would like to see, but you can imagine that AI can also help us finding the right diet in babies with PKU. The study aims for the AI for PKU study are: can we identify factors contributing to the variation in Phe levels in infants with PKU? And with the help of artificial intelligence, we would like to develop a prediction model to predict what Phe will be in infants with PKU and how diet should be adjusted to get the Phe in range. And eventually, we want to develop an AI for PKU newborn application. And the application is not there yet, but here you see how I envision it could look like. So I envision an application in which you fill in relevant information on the baby with PKU, like the age, the weight, the current Phe value, and the current diet. And then the application gives you an advice what to do with the diet, how to adjust the diet to get the phenylalanine in range. So how to achieve our goals? The first important step is to make a database with data of 50 infants with PKU. And in this database, we include data like phenylalanine values, diet, weight, everything that might be important to know. And for this purpose, we will ask people with PKU known in our metabolic centers whether they consent that we use their anonymous data on the period that they were a baby for our database. So this database is very important because this database will be used to train our prediction model. And developing such a prediction model takes several steps. And of course, we will also need to check our results with general statistics. And eventually, when we have a good prediction model, we will be able to develop the AI for PKU application. However, before we can really use the AI for PKU application, further studies need to be done to validate this application. So first, we will make this prediction model in Dutch babies, and you will also need to check whether our prediction model or application also is good predicting Phe and the diet in babies in other countries. But if the AI for PKU newborn application works, it can have a huge impact. It can help us getting Phe in target range soon. It might be able to reduce stress for families dealing with PKU because getting familiar with PKU diet and then having to change the diet many times can be stressful. Also, it can be for an aid for clinicians working with PKU, especially when there are metabolic teams with not a lot of experience or very large clinics. This application might help in getting a good PKU diet in a timely manner. And if this works, this can be a proof of principle for other applications. You might think of an application to get diet advice during pregnancy in PKU. To conclude, with the AI for PKU study, our aim is to develop the AI for PKU newborn application that can guide diet advice to get and maintain Phe levels in range in infants with PKU. I thank you for listening.
Great. Well, thank you. And now I believe she's here live to answer questions. Let's see. Yes. Okay, great. So any questions for, oops, let me just pull up the Q&A. Any questions about this project? Okay, well, one question I see so far, what changes would need to be made for this to be incorporated in the United States?
Thank you for your question. Of course, our aim for this study is to develop the prediction model based on the data of babies from the Netherlands. And I think when we have a prediction model, we should also validate this in other centers, other countries, as for instance, the genetic background may also play a role in the prediction model. So if you want the application to work for all over the world, you also need to validate the application in other populations. And apart from that, you also need to validate the application in real life, that you prospectively test whether it works right, and to really have an application for everyone available, you also have to have all the rules for a medical device. So that will be further steps, further studies, but the first step is to see whether we can make a prediction model, whether we can make this work with help of artificial intelligence.
Okay, great, thank you. And one more question we got. Would the impact of a model like this be less effective if we were to only track and use for a single child? So basically, I think when you were explaining your project, and sorry, I realized my video was off. If you were talking about your project, you were looking at, many people in that healthcare system in the Netherlands. So what would this look like if maybe an implementation, if we're only tracking maybe one or a small number of babies at a time?
Yes, yes. Well, if I understand it well, what you do with machine learning, you use many data to have a prediction model. So to make the prediction model, we will need a lot of data. And of course, a lot of data is for a condition like PKU, which is, well, it's an inborn error of metabolism, so it's not that common. It is difficult to have a lot of data, but we expect with the data of at least 50 infants to have a first prediction model. If you have a prediction model, then you can apply that on a single infant to predict for that infant what the Phe level will be and how you can adjust the diet. But to have that, you will first need the data from a lot of babies with PKU to have the predictive modeling working. I hope that answers the question.
Yes, I think so. So thank you. Okay, so to stay on time, we're going to move to our next one, but thank you so much for answering your questions, and I'll turn my video off. Okay, so the next one will be Dr. Strahosel.
Hello, everyone. My name is Jessica Stroessel, and I'm a genetic metabolic dietitian and postdoctoral research fellow in the Medical Nutrition Therapy for Prevention, or MNT4P program at Emory University. First, I want to thank NPKUA for supporting my research and for giving me the opportunity to share this project with you today. My passion for rare disease research began about 10 years ago when I was an undergraduate student at the University of Wisconsin-Madison. I worked at the Biochemical Genetics Clinic at the Weizmann Center, where I helped a group of wonderful dietitians who cared for patients with inherited metabolic disorders like PKU. This is where I first met families living with these conditions and witnessed both the challenges and the resilience of this community. Those early experiences inspired me to dedicate my career to improving nutrition care for people with PKU. Now in my fellowship at Emory, I combine my clinical work with patients and my research training to ask important questions that can directly improve treatment strategies. Now I'd like to tell you a little bit about our study, which is focused on a very fundamental question, which is how much protein do adults with PKU actually need to stay healthy? We know that protein is essential for growth, repair, and everyday bodily functions. But as you know, for people with PKU, protein management is very complicated because the natural protein that we get from foods contains phenylalanine, or Phe, which must be restricted in the diet. For this reason, many people with PKU rely on medical foods that are free or low in Phe to meet their protein needs. The problem is, even with medical foods, research has shown that growth and nutrition outcomes in PKU are not always optimal. One reason may be that the amino acids in medical food are not used by the body as efficiently as protein from natural foods. This means that people with PKU might actually need more protein than current guidelines recommend. Adding to the complexity of this, PKU is not one single condition. People have different genotypes or changes in their DNA that result in their PKU. And there's also a lot of variation in how much medical food versus the amount of protein from food each person consumes. Despite all of this, the current protein guidelines for PKU are based on very limited data and an outdated method. Our study will use a cutting-edge tool called the Indicator Amino Acid Oxidation Method, or IAAO. The IAAO method is a safe and non-radioactive test that involves giving a person a special labeled amino acid to drink. By measuring how the body processes this amino acid under different levels of protein intake, we can figure out exactly how much protein that person needs. This method has been used in healthy adults, children, pregnant women, and even in smaller PKU studies. What makes our study unique is that we use the IAO method to look at differences by genotype or changes in DNA, which result in mild, moderate, or severe so-called classical PKU presentations. And we'll also consider how the ratio of medical food to natural protein from food a person eats may affect their protein requirements. In other words, we're asking, do people with more severe or classical PKU need higher amounts of protein than those with milder forms of PKU? And does relying more on medical food change their protein needs compared to someone who eats more protein from food? We expect to find that protein requirements in adults with PKU are actually higher than the current guidelines suggest, and that these needs will vary depending on genotype and medical food use. Why does this matter? Because if we can define protein requirements more precisely, we can move closer to personalized nutrition recommendations for people with PKU. This could lead to better health outcomes, improved growth and body composition, and greater flexibility in treatment planning. Ultimately, the results from this study will provide the foundation for updating clinical guidelines so that they reflect the real evidence-based protein needs. Our hope is that this will give both patients and clinicians greater confidence in their treatment decisions and improve long-term quality of life for individuals with PKU. Of course, none of this research happens in isolation. I'm honored to be mentored by Dr. Rani Singh, who many of you may know. Dr. Singh has been a pioneer in genetic metabolic nutrition for over 30 years. She is the director of the MNT for P program at Emory University, and her leadership has shaped the way nutrition treatment for PKU is delivered, not just here in Georgia, but around the world. We are also collaborating with Dr. Raja Alongo and his outstanding team at the University of British Columbia in Canada. Dr. Alongo is internationally recognized for his expertise in studying protein and amino acid requirements, and his team has been instrumental in advancing the methods we will use in this study. And finally, I would like to acknowledge my amazing colleagues in the MNT for P program here at Emory, as well as our partnership with Emory Healthcare and the Georgia Clinical and Translational Science Alliance, all of whom provide critical support for this project. In closing, I want to say how grateful I am to the PKU community for supporting this work. Your participation in research, your advocacy, and your willingness to share your experiences makes studies like this possible. I also want to thank the NPKUA once again for funding this project and for their commitment to advancing research that directly impacts the PKU community. This is a very exciting time for PKU research, and I look forward to sharing our results with you in the future. Thank you so much for listening about our study, and thank you for letting me be a part of this journey with you.
Okay, I think, let's see, hopefully she will come on. Hello.
Sorry, I couldn't hear you for a sec there.
No, no worries. Well, great. Well, anyone have any questions for Dr. Stroessel, please put them in the Q&A. Okay, I do see one. So, if your study does find that adults with PKU need more protein than the current guidelines recommend, what would be the process, the follow-up there to share those results?
Yeah, that's a great question. We already have some preliminary data from our collaborators to show that the recommendations are probably need to be higher than what we currently recommend. So if our study also finds that, we hope that collectively these results will result in revisions in guidelines and ultimately directly into the prescriptions that dietitians recommend to each patient.
Okay, thank you. Let's see if we get any more.
Hi, this is Elena. I was also wondering along those lines, are you planning on doing similar type assessments in children for this?
Yeah, that's a great question as well. It's probably one of our most asked questions is all the populations that need the same attention. I hope that in my career I can repeat the study in almost every demographic of PKU. So kids, it's safe in pregnancy, as well as aging, menopause, etc. So we hope to expand. That's a great question. Thank you.
Great.
Great, thank you. And another question came in. Do you work together with the European guideline group? So would those guidelines, would the European guidelines also theoretically need to be updated as well?
Yeah, so I think the hope for PKU is that eventually there'll be harmony between Europe and America, and that will take a lot of coordination. So for right now, we can't really say what will be updated. We still have to collect all of our data, but hopefully in the future, that's something we can look forward to.
Great, well, thank you.
Thank you.
Okay, and we'll give it maybe another second in case we might need to move on. But another question, which I'm sure many people in the community, are you looking for people with PKU to participate in your project?
Yeah, of course. So we are recruiting adults with PKU. We're focusing right now on young adulthood from 20 to 40 years, just because we know that aging, as well as growth during adolescence can affect protein requirements, and that would kind of give us some results that aren't as accurate as we'd hope. So for right now, just 20 to 40, but there'll be future studies as well in older populations.
Great, well, thank you so much. That was very helpful. Okay, so we'll go ahead and move on to our next video. So I'll turn my video off, and we will hear now from Dr. Weisbrunn.
Hello, I'm Susan Weisbrunn, and let me introduce myself a little bit. I'm a psychologist, and I've worked at Boston Children's Hospital for the last 45 years within the clinic. About 5-6 years ago, I retired from clinical work, but continued to do research and some consulting to pharmaceutical companies developing new treatments for these rare metabolic disorders, including PKU. Today, I'm going to talk about a project that was recently funded by the NPKUA called Proficiency in Verbal Discourse, a proposed outcome measure for clinical trials in PKU. The overall objective is to correlate changes in verbal discourse with changes in blood Phe in the same person. This approach aims to validate a linguistic marker that could speed up drug development and possibly improve management of PKU. My collaborator in all of this research is Guillermo Ketchy from IBM Watson Research Division of Digital Health. So I'll speak about the following things today: a brief background of the verbal discourse types of research, a summary of our first study, also supported by NPKUA, and an overview of the current study. How the cookie theft picture came to being our focus. A long time ago, maybe almost 6-7 years ago, I noticed that when individuals, adults with PKU, were on pegvaliase or other treatments to lower their blood phenylalanine levels, their verbal discourse improved dramatically. So I started looking up tests that might be used, and I found the cookie test. And then I found an article by Guillermo Ketchy and his colleagues showing that using the cookie theft samples of language, in which people simply described what they saw in this old-fashioned picture, that they could identify declines in language in people who eventually became demented with Alzheimer's disease 10 years before any traditional neuropsychological tests could identify that. So I called him up and asked if he could identify benefits or improvements in language. And he said he didn't know. And of course, he'd never heard about PKU, but that if I wanted to do a study, he would analyze our data for free. And such began the study. So our initial primary study hypotheses were that the cookie theft picture task would be useful for this kind of study, also using remote administration, since this was during COVID. And also that it could identify linguistic markers that could differentiate people with PKU from a non-PKU group, because first we had to show that there was a difference, if not a deficit, and that we could correlate this marker with blood Phe levels on the same day that they were taken. So that we found that the PKU group did differ significantly from the non-PKU comparison group. more importantly, we saw when we looked at people who scored high on this dimension and people who scored low on this dimension, that it correlated highly significantly with the blood phenylalanine levels. Those who had low phenylalanine levels scored high, and those who had low phenylalanine levels, scored high, and those who had high phenylalanine levels scored low on this dimension. Then we had to know, what was this dimension? So we gave 2 samples to ChatGPT to say, what's the difference between these samples that scored high on this measure and those that scored low on this measure? And lo and behold, it came back with a description, basically executive functioning aspects, complexity and depth of description, detail orientation, the narrative coherence. In other words, that they followed along the picture. They would describe one part and then the next part. And the last aspect was recognition of emotion, which I found extremely interesting. We published this study earlier this year and decided that the next step we really had to do is, could this dimension, which we called proficiency in verbal discourse, also be sensitive to changes in blood phenylalanine levels in the same person when it was administered over time. So the specific aims of this study, which was recently funded, is to confirm that this proficiency in verbal discourse score distinguishes adults from PKU from a healthy comparison group, and that there is this within-person change correlates with changes in blood phe. And finally, that this score reflects self-reported perceptions of functioning and quality of life, but it represents a distinct aspect of language that does not correlate with traditional measures of language. So this study will involve a follow-up using the same participants from the last study. And it'll involve 2 remote sessions over the secure BCH Zoom, that's Boston Children's Hospital. These sessions will be at least 2 months apart, and the total time for each session is 45 to 60 minutes. Participants with PKU will receive a $50 gift card, and the comparison group will receive a $35 gift card. Everybody will receive a summary of the study findings, and we will also publish it. So the participants will be recruited from our previous study. For the current study, we'll only be using 20 from each group due to the fact that we'll be seeing each one twice and the financial limitations. As you can see in our previous group, they were about the same age as the comparison group, mean age around 36, 38. They were highly educated, 79% had gone to college. Their verbal comprehension index, which is considered a verbal IQ, was not different from those in the comparison group. An average score is 110, so you can, 100. So you can see that this is, they even scored above the population norm. However, when they did the self-report of executive functioning, they were significantly higher. And this measure, the brief, high scores indicate having difficulties in planning, organization, memory, information processing, that kind of thing. And 26% of them were actually in the range of a significant deficit, compared to only 7% in the comparison group. Only 13% had ever gone off diet, and the mean blood phenylalanine level was pretty close to the recommended ranges, though the range was quite high. What will we be using to study this? We have a background questionnaire developed for the study that gives us information about their education and marital status and work. We'll use the cookie theft picture task again, but at the recommendation of our colleagues at IBM, we're using a less structured language sample as well, just simply asking, how are you feeling and why? We'll be giving parts of the WAIS-4, which is the standard IQ test, which will allow us to have a verbal comprehension score, which is the verbal IQ, as well as a memory score. We'll be repeating the behavior rating inventory of executive function. That's the executive functioning test that we did before. That's a self-report. And we're also going to be having them complete the patient global impression scales. And this is just, how are you doing now? And then at the end, how are you doing after for the second time? Has there been a change? And finally, for the PKU group, we'll obtain a filter paper blood specimen at the same day as the study. And remember,
Sorry, I think it.
Remember, this is all on Zoom, but we actually will watch them take a blood specimen so we know that it was done on the same day. So the timeline is that we'll have 80 study sessions because each participant will be seen twice. So we will have 3 data points because we'll have the old study and then the 2 new ones. And in the first months, we'll complete our administrative tasks and begin recruiting and enrolling subjects and complete the first 20 study sessions, 10 from each study group. Months 4 to 6, we'll conduct 30 more study sessions and we'll submit our interim report on February 26th. And months 7 to 9, we'll complete the final 30 study sessions. And at the end, we'll complete the data entry, submit language samples to IBM for analysis, prepare our manuscripts for publication, and a lay summary. And I forgot to mention, one of the key things about this study is that this measure is a single number. It's unbiased because it's based on performance, not self-report. And we believe that it will correlate with the self-reported aspects, how they're feeling on the patient-reported outcomes, but that it really is something unique. And that's kind of what we hope to get out of it. So I want to thank you for the opportunity to pursue this study and hope you can capture how excited I am to be able to study this. Thank you. Hello.
Okay, great. So now we will move on to questions from Dr. Weisbrunn, who I know is here. Let's see, Dr. Weisbrunn, are you still here and able to hop on video?
There. Okay. Yep. And let me
Okay.
get on video too.
There we see you. And if anyone would like
Can you see me now? Oh, good. Okay.
Yes,
Yep.
now we can see you.
Okay.
If anyone would like to jump in the Q&A and ask any questions. And I'll start with one similar to the last one.
Okay.
Are you looking for any other people or any other community members to participate in this project? I know you mentioned that you are going back maybe to the people from your previous project, but any other comparison group or any other community members that you would like to participate in this project?
Well, if we don't reach our intended sample sizes, we would love to have other people participate. Since we're doing it twice, we will still have reasonable data to answer our questions. But right now, no. But we are also trying to organize future studies that will include children as well. And then we will definitely be reaching out again to the NPKUA, I'm sure, for recruitment, if not funding.
Great. And Alina, do you have a question?
Yeah, so Susan, that was a great presentation. Thanks for the update. How do you envision this type of test being implemented in an investigational clinical trial to, for a new treatment for PKU?
Well, as we've been working on this, and as I've been working with the pharmaceutical companies who are interested in this type of a measure, I realized that with the FDA, they won't accept it as a validated measure yet, but it will be exploratory, one of the exploratory measures, and it could support the findings. The importance of it is that it's a functional, objective measure rather than a patient-reported outcome. While the patient-reported outcomes are important, often they're not strong enough for the FDA to say, oh, that's enough. They want to show that it really does have an impact on day-to-day functioning. And it makes sense to me that they say that, and that also that this measure can do that. So we're hoping that through this study, we can have enough background data to support its inclusion in clinical trials.
And do you ever envision this being able to be used in clinical practice?
In the future, possibly. Right now, it's a research tool. The same issue that was talked about before. How do you incorporate AI into the community, into community medicine, clinical medicine? It's going there, but it isn't there yet.
Great. Thank you so much. We just want to make sure we have time for our last video, so we'll go ahead and move on to that one. But thank you, Dr. Weisbrunn, for joining. Okay, so now we will hear from Dr. Amir Zarnpar.
Hi, everyone. My name is Amir Zaranpar, and I'm a professor of gastroenterology at UC San Diego. Among the many different hats that I wear, I am the co-director of the Center for Microbiome Innovation, where my colleagues and I are actively thinking about how we can use the microbiome to treat a variety of diseases. It is such a pleasure to be one of the NPKUA award recipients this year. This is the second time that my lab has received an award from NPKUA, and we're hoping that it will help extend some of the early work that we did with partnership with the NPKUA. Let me tell you a little bit about our project. When you look in the mirror, you're not only looking at your human self, but you're also looking at trillions of bacteria that live on top of and inside of you. In fact, when you count the number of cells that are in your body versus the number of microbes that live in and on top of you, it's a 1-to-1 ratio. So really, you're only looking at a meta-organism that's made up of so many different species, of which half of all the cells belong to you. The important thing about the microbiome is that it is extending the genetic toolbox that we have. Whereas our human genome has only about 46,000 genes, 20,000 of which encode proteins, our gut microbiome or oral microbiome bring to the table 46 million other genes that are at our disposal and for our use. In fact, the way that we think of the microbiome is that it's a portable toolbox that can adapt to any situation. Our microbiome in our gut adapts to the kind of food that we eat and helps us digest the different kinds of foods and be able to get different kinds of vitamins and so on and so forth. Our skin microbiome helps us adapt to the different climates that are out there as well, or the different kinds of clothes that we wear. So all of these microbes are performing important functions that are human cells rely on for health. And when you think of the microbiome as being a potential toolbox, it really expands the ways that you can potentially use the microbiome for health. Since the microbiome has such a huge influence on nutrient absorption and our immunology and so on and so forth, we think of it as a way that we can potentially treat a myriad of disorders. And because we have a microbiome on basically any surface of our body, it has a lot of different access points, so a lot of different ways that we can use the bacteria for a variety of diseases. And importantly, if we figure out how to get these bacteria that we manipulate to help us improve our health to become part of the ecosystem, then these bacteria could have potentially curative potential. And I think that PKU is a potential disease in which we can have a really big impact like this. But why hasn't this worked already? Well, the microbiome therapies do not work because, or have not worked, because the microbiome between me and my spouse, my colleagues, someone who lives in Europe, someone who lives in Japan, for example, all of our microbiomes are so vastly different from each other. And not only that, not only are our microbiomes different from each other's, my microbiome today is going to be different than my microbiome tomorrow, and it's different from my microbiome from 2 years ago, mainly because of the diet and things that I'm eating. My microbiome is constantly changing and adapting to the different environmental things that they're being exposed to. So when you think of potentially having an engineered probiotic go in and do something beneficial, well, this bacteria is going to have a really hard time competing with these bacteria that have grown inside and on top of me for my entire life, basically. These bacteria that are probiotics have a really hard time trying to find a place where they can survive or find the nutrients that they need. And so they don't survive long enough to do anything beneficial. And even if they do survive, they're not, they're spending so much time trying to figure out how to survive that they're definitely not going to do something that I want them to do, like cure PKU, because they're not they're only doing the functions that's trying to help them survive in these hostile environments. So the way that we have addressed this problem is to think really deeply about what are the bacteria that we're using to introduce functions. And by the bacteria to introduce functions, we call those the chassis. It's kind of like the same way that you have a chassis for a car, this frame on which you can build stuff on top of. Well, in this case, what is the bacteria in which we're putting these beneficial functions in to try to treat something like PKU? And you know, when you look at the different bacteria that have been used to treat PKU in the past, E. coli Nissl, which Synlogic famously used, or Lactococcus lactis, these bacteria have never survived or never been in a human being. They're not going to be able to go into these environments and survive long enough to do something beneficial. It's like throwing a pet hamster into the Amazon jungle. There's just going to be too many other, you know, it's going to have a hard time finding food or competing or being able to survive all these various predators that are there out to destroy it. Same way these bacteria aren't prepared to be in that area. So we were thinking deeply about, well, how do we identify a chassis that can be strong enough to engraft or stay in the gut to do something beneficial, but be easy to contain. And the solution that we came up with was that instead of picking a bacteria that's never lived in the gut before, what if we picked a bacteria that was already in the gut, that was already part of the community, that was already really well adapted to be part of the system? What if we took a bacteria that's already there and engineer it to do something really beneficial to help the host survive? That's really what the innovation in our lab is, to identify bacteria that are part of these ecosystems and engineer them to do something beneficial. And we've actually been able to show that when we use a bacteria that's already part of that system and engineer that, we can get these bacteria to become part of the community and stay within that mouse. We've only done these experiments in mice, but stay within that mouse for the rest of its life and perform these functions for the rest of the life of the mouse. And we've been using this in primarily diabetes. We show that we can improve prevent diabetes, basically lower blood glucose in these mice for their entire life with just a single treatment of a bacteria that's expressing a beneficial function. Importantly, these bacteria also like to live in the small intestine, which is where a lot of digestion occurs. So they're able to do a lot of these functions that we knock into them earlier or in the gut, which is really beneficial for something like PKU. Well, the question you may be asking is, well, have we engineered these bacteria to metabolize phenylalanine in a way that could help a patient with PKU? And that was what the first grant that we received from the NPKU did. It helped us build these bacteria that, as you can see here, are able to decrease phenylalanine and increase its byproducts by having a number of copies of these of a phenylalanine metabolizing gene. What the grant this year will do is it will help us put these bacteria into mice and see whether they stay long-term in these mice and are able to treat PKU in these mice. We're really excited to see if this works, and we really think that it's going to be impactful. Those of you who may be familiar with Synlogic, with a bacteria that wasn't able to engraft, that really struggled in that environment, a company was able to go to phase 3 trials. We're hoping that by using a bacteria that can survive long enough, we'll be able to succeed in our phase 3 trials. But first, we got to see if this works in mice so we can get approval to do this in humans. This is the team of people who will be doing these experiments, the people in my lab. I also like to thank Carrie Harding in Oregon Health, who's been helping us and advising us on how to do these studies in mice that have PKU. As you can tell, we're not a rare disease lab, we're a microbiome lab, but we think that this is a great opportunity, and we are excited to see if we can help patients with this disease. Thank you.
Great. Well, now we will do some quick questions. I know we are over time, so we will see if we have any. Thank you, Dr. Zarenpar, for being here. And let's see, it looks like we have something in the Q&A. We do have one question more, and I know you alluded to this on your in your presentation, but could you just explain a little bit more about how this study will be different than the Synlogic? And I know you mentioned with the different bacteria, but if you could elaborate on that.
Sure thing. Synlogic used a type of bacteria that's been known to be safe. Mainly, it's been used in the lab a lot, and it's very easy to engineer. But the problem with using a bacteria that's living in a lab as opposed to living in a person is that that bacteria isn't really adapted to live in our gut. Our microbiome does a lot of things, but one of the things it does really well is prevent other bacteria from getting in, and that's what the bacteria that Synlogic used. That was what it was struggling against. So they kept on having to increase the dose, which then caused a lot of nausea. And of course, if the bacteria is trying to survive, it's not really doing the things that you want it to. But what we're doing is that we're using a bacteria that's already part of that ecosystem, that already belongs in the gut. The trade-off is that it's much harder to engineer. These bacteria, because they haven't been in a lab and haven't been selected for being easily engineerable, they're harder to modify. But thanks to a lot of innovation that's gone into this space in the last 10 years or so, we've begun to be able to modify these, what we call undomesticated bacteria, to do things that we want them to do. And when we put these bacteria then back into the ecosystem that they belong, they can go back to their home. And now, since they're not really struggling to survive, do all the things that we want them to do. And in this case, hopefully metabolize phenylalanine in a level that improves patient health.
Great. Well, thank you so much. I know we are over time, so I think we will have to leave it at that. But we will be sharing the recording of this webinar. So if you want to go back and watch these videos and ever have other follow-up questions, I am sure all of our grant awardees would love to answer any questions after this. So thank you all so much for your attendance, and we hope you will join us for future webinars. So thank you.